In this video we're going to be talking about biochemistry. Now in biochemistry we have lots of little things that we have to know, but not in too much details. But one of the things that are fundamental in understanding the basis of biology is enzymes. What are enzymes? Enzymes are biological catalysts. These guys are considered as proteins. These enzymes that are biological catalysts are proteins. They fall under the category of proteins. Every enzyme is a protein. And this enzyme is a catalyst that is going to break something apart. That is going to break down a substrate. Let's keep it simple. It's going to break down a substrate. And how it does that and how it speeds up, oh by the way, enzymes speed up a reaction. Enzymes speed up reactions. And without enzymes, really, without enzymes, you cannot survive. Enzymes allow our body to carry out reactions at such a fast-paced manner. And it does this by lowering the activation energy. Enzymes lower the activation energy. And it's required to break down this substrate or to break down this compound. So let's look at some enzymes now. We're going to keep it simple and then slightly make it advanced. So enzymes, very, very cool thing. These are proteins, and enzymes, they are remarkable. I mean, these guys are in every single reaction, and these enzymes lower the activation energy and start to break down something. So let's pretend, let me just draw a very simple enzyme right now. Here's my enzyme. And this enzyme is going to be called lipase. Anything with a ase ending refers to an enzyme. Ase ending, suffixes that have ase refer to enzymes. Lipase. Lipase means enzymes that break down fat. Lipids. Lipids are fat. Lipase is the enzyme that breaks down lipids. How do you break down a lipid? Well, one lipid will essentially turn into one glycerol plus 3 fatty acids. And going on this notion, let's draw something here. Let's draw a few compounds. We can draw some triangle over here. I can draw some weird looking shape over here. And this, I'm not going to spend too much time because even a second grader can understand that obviously this guy, this substrate over here, has to fit in here and not these two. These two are not compatible with this. What do you call this right here? This is called the active site. This right here is called the active site of the enzyme. Active site, and the active site is where the substrate will bind onto. Here is my substrate. So, substrate will bind onto the active site and break it down. And now, to have a visual description of what happens, perhaps you've taken this substrate, and now you have a bunch of these little square molecules. So this really gives you a visual description of what happens. Let's keep it simple. Lipase breaks down lipids. You can also have other enzymes. So, for example, protease. Protease breaks down protein. And you have a bunch of enzymes, but understand lipase, protease, amylase breaks down starch. So I think these are the three of the enzymes that you should know in the back of your mind. And whenever you see a crazy enzyme, something like amino acyl tRNA synthetase, which you do have to actually learn when we talk about molecular biology, but just learn it's an enzyme that does something. But anyways, okay, so I understand. I have my active site, I need the substrate to bind onto the active site, and then my enzyme will break it down. Okay, great. We have a model for this. This enzymatic model has a model called the Induced Fit Model. So the enzyme, how the enzyme works, it's referred to as the Induced Fit Model. The enzyme follows a Induced Fit Model, and basically here's what it states. Several years ago, teachers used to teach students that the model for enzyme is a lock and key model. That is very wrong. Nothing is perfect. No substrate is going to fit in perfectly to the active site. That is what the lock and key model states. Remember, lock and key model is wrong. The enzyme follows something called the Induced Fit Model. Here's the Induced Fit Model. Basically, here's my substrate, here's my active site. And my active site is going to slightly, not a lot, it's not going to change a lot, but it's going to slightly change in shape to accommodate the substrate. So it's not moving like this, it's moving perhaps like this. And it's going to accommodate my substrate and then break it down and break it down. Remember, no substrate is specific in size and how it looks. So the active site has to slightly conform in shape to get a good grip on the substrate and break it down. This is called the Induced Fit Model. Please remember that. It's not lock and key. It is not the lock and key model. So moving forward, enzymes, before we talk about how they're regulated and how they work best, let's talk about allosteric sites. Enzymes may have an allosteric site. What does that mean? Well, an allosteric site is a site other than the active site that is present on the enzyme. Here is my allosteric site. It's not my active site, it's somewhere else. And the allosteric site allows for non-competitive inhibition. It's very cool. You're going to learn, we're going to talk about non-competitive inhibition versus competitive inhibition. Non-competitive inhibition, meaning there's no competition, is when there's an allosteric site present on the enzyme, and if a molecule binds on here, this allosteric site can either repress this entire thing, meaning the active site will no longer work to break down whatever it needs to break down, or this enzyme can only break down substrates if the compound is attached to the allosteric site. So it can work either as a repressor or an activator. Make sure you remember that. It can either work as a repressor or an activator. Non-competitive inhibition. You simply bind the active allosteric site and that's it. Your active site will no longer break down whatever you have to break down. This is called non-competitive inhibition and it uses the allosteric site. Important to know. Now, what's competitive inhibition? This is where molecules or substrates compete for the active site. For the active site. So what does this mean, competitive inhibition? This means that molecules that resemble this specific substrate right here are going to try to get in. Now hold on, why do we have things, why did I draw this in two different colors? We're talking about competitive inhibition. You might say, hey, why do we want to inhibit stuff? Right? Well, let's give you a real life example. Let's talk about aspirin. What does aspirin do? It reduces headaches, it reduces the symptoms of fever and pain and such and such. Aspirin mimics the substrate that is required for an enzyme called cyclooxygenase. So let's just say here's an enzyme, and this enzyme is called cyclooxygenase. Do you have to know this? No, you don't. If you want to be a nerd, if you want to know some more cool stuff, maybe some pharmacology, basic pharmacology, memorize it. It's not hard. Cyclooxygenase. It's an enzyme. When the substrates bind onto it, guess what? It activates the pain pathway. It actually activates prostaglandins and thromboplastins. But let's just say prostaglandin pathway promotes pain and such and such. Cyclooxygenase promotes pain pathway, promotes all these symptoms that you're feeling. But what does aspirin do? So you have these substrate molecules that will cause you pain, cause you to feel fever, and aspirin essentially is a molecule that just looks exactly like it, but it mimics it in shape. Sorry, that doesn't exactly look like it, but it mimics in the shape. And the thing about this is that in aspirin, we have a lot more of these compared to the actual molecules. So now we can have a nice competitive inhibition. The ratio of these are much higher than these ones, so guess what? The chance of these binding onto the cyclooxygenase are much more likely, and they will inhibit. They will not allow the enzyme to activate this pain pathway, and now you're going to start seeing reducing and swelling and other things, and the prostaglandin pathway will be inactive for a period of time. And this is called non-competitive inhibition. You have to compete for these substrates to fit into the active site. Whereas in the non-competitive inhibition, you have allosteric site. If something attaches here, that's it. It's a done deal. There is no competition going on here. This is very, very important for you to understand. Non-competitive inhibition versus competitive inhibition. This is just one example. Very important. So there you have it with non-competitive inhibition and competitive inhibition. Understand allosteric sites, understand active sites, very, very easy. Enzymes lower the activation energy of a reaction, they speed up the process. And now let's talk about the last two factors that I want you to understand about enzymes. Enzymes are affected by two things. Two factors that affect enzyme activity. This is very important if you're taking the Regents, by the way. Two factors, or the biology state test. Two factors that affect enzyme activity are temperature and pH. Temperature and pH. Temperature and pH. Very, very, very important. These two factors will determine the rate of reaction, the rate of enzymatic activity. What do I mean by that? Well, there's a lot of enzymes, and each enzyme has an optimal pH, and an optimal temperature. So let's give an example. Let's pretend we have an enzyme called trypsin. And trypsin is an enzyme that's found in your small intestine, and it actually has an optimal pH of about 8. You should memorize this. You should also memorize pepsin. Pepsin and trypsin you should know. Pepsin is found in the stomach and has a optimal pH of about 2 to 3. It has an optimal pH of about 2 to 3. Trypsin has an optimal pH of about 8, located on the small intestine. Let's try to draw a graph of what would happen. Let's work with pH first. So here's my graph, and here's my pH. Here's my rate of enzyme activity, or rate of enzyme activity. Let's see what happens. Well, pH scale goes from what? 1 to 14. So I'm not going to label all of them, but perhaps I'll just label a few. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. This is 14, 4, 5, 6, 7. This is 8, this is 9. This is 13. Let's see what happens. And of course, this is increasing. What is going to happen? How would the curve look for trypsin? Well, trypsin works best at a pH of 8. Well, let's see, pH of 8, that means there should be a peak. The highest peak should be at a pH of 8. And as we increase the pH, the enzymatic activity will decrease. It will decrease. As we decrease the pH, it will also decrease. So, what is the optimum pH level of pepsin? Well, let's see. It's 8. If they give you a graph and they say, what is the optimal pH of this enzyme? You just find the highest peak and you say, hey, look, it's 8. What happens as you increase the pH level or decrease the pH level? Well, you are denaturing your enzyme. You are denaturing your enzyme. Write this down, very important. You're denaturing your enzyme. Denaturization. Denature. You are changing the active site. The active site. Very, very important. Denaturization means that you're changing the active site of the enzyme. If you change the active site of an enzyme that is supposed to break down things like this, and it ends up looking something like this, well, this is denatured. This enzyme cannot appropriately now break down the specific substrates it was supposed to break down. And the pH changes the conformation of the active site. It messes with this tertiary stage of this enzyme. And that's it. This protein is basically gone. So high levels of pH, low levels of pH, no good. We have an optimal amount of pH, and then of course, if we make it to 9, it'll work, but not as efficient. Easily, since we did trypsin, now we should be able to do pepsin by yourself. Hopefully you realize pepsin has about a pH of 2 to 3. I'll draw it over here. And it should look something like this. Once again, if something tells you, hey, what is the optimal pH of pepsin, it should be 2 to 3 over here. So here, it's an error. Pepsin goes here. I don't know why I labeled this pepsin. Hopefully you guys caught on. This is trypsin. Trypsin is found in the small intestine. Hopefully I wasn't saying that throughout the entire video. This is trypsin. Trypsin is found in the small intestine, has a pH of 8. Pepsin has this pH of 2 to 3. So this is what the graph of pepsin should look like. This is what the graph of trypsin should look like. And this is basically, this is a very, very familiar question on the Regents examination. Not typically the AP. This is a bit too easy for the AP examination. And now the last factor, let's talk about the temperature, and we'll conclude this video. So, in the temperature, how does temperature affect enzymatic activity? Well, let's see. Temperature. If I increase, let's make it simple. If I increase my temperature, the rate of the enzymatic activity increases as well. Enzymatic increases. However, once you increase the temperature too much, you will cause denaturization. You will denature your enzyme if you increase the temperature too much. It's a very important idea to distinguish. If I increase my temperature in an enzymatic reaction, now enzyme activity will increase to a point, and then the enzyme will start to denature because it's way too hot. So remember this. First it increases, and then it will denature, and the enzyme no longer works. It's non-functional. Interestingly, on the opposite case, if I decrease my temperature, the enzymatic activity slows down and eventually stops. It does not denature. Please make note of that. A lot of people confuse it and just remember that it's denature. pH increase, denature. pH decrease, denature. Temperature increase, denature. Temperature decrease is not denaturization. Nothing happens. You just freeze the enzyme. The enzyme no longer works. However, if you bring it back up to room temperature, or if you bring it back up to the optimal temperature that the enzyme functions in, then the enzyme will work again. How do you think we do it in the lab? Later on in the video, we're going to be talking about genetic engineering and molecular biology, and we use enzymes, restriction enzymes, or just any enzymes in general, but when we don't use them, we have to refrigerate them in a very, 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 very cold setting. And then when we want to use them again, we can now bring them back into room temperature. So understand the relationship of enzymatic activity between temperature and pH. And that is basically it. This concludes the video for biochemistry enzymes.